So guys, in this section we will be going through the structure of a prokaryote, right? So in case of a monera, what we observe that there are bit of prokaryotic structure. Prokaryotic means they don't have any kind of developed nuclei, right? Suppose this is a bacilli cell, and now give it glycocalyx. Then we will have another layer. Just draw it in freehand, right? There are certain mesosome structures. These mesosomes helps in the binary fission of bacteria. Then we will have a nucleoid, right? And afterwards, from here, we will get a beautiful flagellum. And this flagellum works in a particular direction. So they turn in a particular direction while the bacteria goes for chemotaxis. That means due to some chemical signals, it can move from one place to another, right? Chemical stimulus play a role in this case. Then we will have certain amounts of granules, right? And after these granules, we will be able to observe certain cilia in the three-dimensional structure and certain large structures. These structures that develop from the membrane, cell membrane, these are known as pili or fimbriae, right? These are known as pili or fimbriae. Then we will have some short stretch of RNA, certain amount of enzymes in here, then certain amount of polyribosome. They will show certain amount of polyribosomes, then mitochondria for the energy, right? So these are the basic organelles that we will be able to find in a particular bacterial cell. Now let's see how we can go for these structures, why we will be labeling them, right? So the total structure in this case outside the cell membrane, this total structure will be known as glycocalyx. Afterwards we will get a cell wall, and the inside we will get the cell membrane. As is do not have any kind of developed nuclei, only the nucleoid flows in here. This is known as nucleoid. Only the nucleoid flow in here. Then we can have this portion of cell sac or hyaloplasm or cytoplasm. The total portion will be known as protoplasm, as it do not have any kind of developed nuclei, right? Okay, so these are cilia. These cilia helps in the propagation in different directions, right? These are known as fimbriae or pili. These helps in the conjugation process of bacteria. This helps in the conjugation process of bacteria, right? This is known as flagellum. This gives the propulsion effect. This gives the propulsion effect for a bacteria to move forward or backward. Now in here, what we have seen, there are lots and lots of ribosomes in an mRNA. So it will be known as the polyribosome. This will be known as the polyribosome, right? The polyribosome, and afterwards, let's zoom it. That's it. Okay. And afterwards, in here we need energy, right? That's why we require some amount of mitochondria. And now there are certain amount of free floating RNA that are the messenger RNA. They are, what should I try? In here, these are mRNAs or messenger RNAs in this particular section. Certain amount of biological enzymes. There are certain enzymes. This specialized structure that helps, what I have told you earlier, that specialized structure mesosome is that helps in the binary fission, right? It holds the nucleotide in a particular section. So we will go through the different functions of these structures later. So this is known as the mesosome, and what we will get after these things, we will get a short stretch of DNA molecules. There are shorter stretches of DNA molecules. So whenever it comes, in case of NCERT books or NEET or ICAR, what we gonna learn in the CBSE subjects, we will find these structures, right? So that was all about prokaryotic structure and how we will be able to draw that structure. This profile explains the structure well, right? Now let's, when we know that a bacteria look like, so how does a cocci looks like in this type of figure, right? So let's see. What I have told you, that cocci is a spherical shaped bacteria. So this will be a spherical shaped bacteria. Suppose I am drawing this kind of particular structure, right? So in case of spherical shaped bacteria, we will have certain amount of certain structures related to its functions, right? So in case of pneumococci, we will get a particular structure. These are basically pneumococci. Pneumococci are basically streptococci bacteria. Pneumococci. These are basically known as the streptococci bacteria, right? Streptococci bacteria. So streptococci basically have a structure regarding their structure. So they have a particularly delicate structure. Actually what happens, they form a literally balloon shaped structure, and they look like this one. They form a long chain of structure, right? This causes pneumonia to our body. So let's see what are the different kinds of structures that we can find, or organisms we can find in a particular pneumococci, right? Let's see. So in case of a pneumococci, what we will be having, that they will have a structure like this one, and the outward structure will be like this one, right? Then what I have told you, that there will be certain amount of M protein or fimbres will be there, and lipo. There will be a lipoteichoic acid, right? So this one, all these are known as fimbi, and then we will have a beautiful nucleoid in this here, in this section, and afterwards there will be a lipoteic structure in here. Okay, so let's see, this is the cell wall. This will be known as the cell wall. These are known as M protein of fimbriae. These are known as the M proteins of fimbriae, and afterwards we will get certain number of proteins that will be in here. These proteins includes three kinds of proteins, that is, number one, invasins. These invade in the body of the host. Number two, this was one, this is two. Number two will be the streptolysin. They show, these kind of proteins show certain amount of lysozymic activity, and that lysozymic activity leads to the invasion into a particular host body, and then we will go for the streptokinase. These streptokinase actually, they deal with the glycolysis of the host body, right? They disrupt the regular respiration process in a particular host body, and afterwards we will be able to find some proteases. These proteases will cleave the protein, and then that protein will be hydrolyzed into certain amount of amino acids. Okay, so we will be looking there the different structures of the protein later, but now in here we will get the inner part known as the plasma membrane. This is known as protoplasm, and afterwards what we will get, that there are certain stronger structures that holds the whole glycocalyx region. This one, mycelial mucilaginous structure, or they will be known as certain proteins known as adhesins, right? There will be certain proteins known as adhesins. These adhesins will help to adhere to a particular host body. Okay, so these structures, these three structures, number one is the M protein, number two is lipothalic acid, and protein F and TB. All these three structures will form the protein known as adenosine, right? So all these structures will together form a particular kind of pneumococci structures. These pneumococci are associated with the respiratory diseases in case of mammals, right? So what we have learnt earlier, that this lipoteichoic acid will always form a particular structure that will help to make that adenosine. Now, how this particular adenosine works? Suppose there is a particular cell membrane in here. Now, this virus will have these three kinds of acids. Suppose this is lipotylic acid, this is M protein, and this is the Pro-F in this case, right? All these will form the adenosine that will help to adhere that particular pneumococci in a particular host cell. They will go and bind into a host cell and will help to adhere that particular host cell to its own body. So that's all about the pneumococci. Just subscribe and